Good afternoon. In this video, I'm going to discuss an introduction to biochemical engineering. Basically, we're going to take a look at the biochem industries and also the process involved in designing a biochem industry. So let me share the PowerPoint. Okay, so biochemical engineering, this is the application of chemical engineering principles to the processing of materials using biological agents such as cells or enzymes to achieve a desired chemical transformation on an industrial scale. So basically, biochemical engineering is the scaling up of the laboratory methods of the biochemists or the geneticists or the uh, microbiologists, no, to industrial scale. So biochemical engineering is a component of biotechnology. So generally there are four components of biotechnology, you know, microbiology, biochemistry, genetics, and then the fourth one is biochemical engineering. Okay, so biotechnology, on the other hand, this is the practical application of biological agents. So biological agents can be living or dead cells or subcellular components like enzymes, ano, in technically useful operation. Either in productive manufacture, like for example, we're going to produce ethanol, no, service operation or environmental management. So for example, we're going to treat wastewater. So that's environmental management. So these are the different biological agents, microorganisms like bacteria, yeast, fungi, algae, no, or plant and animal cells, subcellular components like enzymes. So these biological agents are involved in biochemical engineering. Technically useful operation. So technically useful operation refers to the manufacture of economically useful or valuable product like single cell protein. So ito yung we use bacteria to produce protein, and then we use the protein in animal feeds. Okay naman, pharmaceutical products, no? Like antibiotics, we use penicillium yeast to produce penicillin, gano'n, ano? Industrial chemicals like ethanol. So, and also the waste treatment is also part of biotechnology, biochemical engineering. So these are the list of products produced by using biochemical engineering, and also the organism used to produce this product. So for example, ethanol is produced using Saccharomyces cerevisiae and also the approximate world market. So as you can see, pinakamalaki talaga dito is ethanol. It is produced at, what's this, 20 million, no? 20 million tons per year. And then acetone and butanol is produced using Clostridium acetobutylicum at 2 million tons per year. Ganong kalalaki itong production na to. Ano? And then we have the biomass. Biomass single cell protein. You can use Candida utilis, Pseudomonas, Baker's yeast. No? This is at 0.5 to 1 million tons per year. And then we have the organic acid, say citric acid. Citric acid is produced using Aspergillus niger at 2 to 3 million tons per year. No? And then lactic acid. we're using Lactobacillus delbrueckii at 200,000 tons per year. So sometimes kasi you need to memorize the organism and the product. So sometimes the organism sounds like the product. So for example, in lactic acid, the organism is Lactobacillus delbrueckii. Uh, Amino acids, these are the different amino acids. For example, glutamate. This is the glutamate in your betsin. So it is produced using Corynebacterium glutamicum. So basta tandaan nyo lang naman, multiple choice naman sa board exam. So some organism sounds like their product. Okay, so this is produced at 3 million tons per year. And then lysine, if you still remember, this is the amino acid that is generally used to improve the appetite of children. This is usually incorporated with multivitamins and mineral preparation. And this is produced by Brevibacterium flavum. And then phenylalanine. Phenylalanine is produced using Corynebacterium glutamicum. The same bacteria that produces the glutamic acid. Next, we have the exocellular polysaccharides. Ibig sabihin, these are produced outside of the cell of the microorganism. So for example, santan gum is produced using Xanthomonas campestris, and dextran is produced using Leuconostoc mesenteroides. Proteases is produced using Bacillus sp. So this is an example of an enzyme. Proteases, if you still remember, this is used to produce white cheese, yung kesong puti. And then amylases, this is produced using Bacillus amyloliquipazins. And then pectinases is produced using Aspergillus niger again. So vitamins, for example, vitamin B12 is produced using Propunicum sermani at 10 tons per year. And then pigments, we produce beta-carotene using Blanklesia trispora at 60 kilogram per year. Usually, beta-carotene is used in the production of infant formula. Ninadagdagan ng beta-carotene yung mga gatas ng baby. Chikunine is produced using Lestospermum erythrorhizon, a plant cell. Okay? So, and also used to produce vaccine. So for example, tetanus, this is the drug for this is the anti-tetanus vaccine, is produced using Clostridium tetani. The hepatitis B vaccine is produced using surface antigen in recombinant yeast. So in the future, pag meron na tayong industrial scale ng COVID-19 vaccine, alam niyo naman, this is produced using RNA, di ba? RNA is a part of a cell, yan. Tapos pag yun namang, yung mga yung sa AstraZeneca, ganito pa rin yun, di ba? They are producing vaccine from closely related bacteria. Closely related virus, Closely related virus. Like AstraZeneca, di ba? It's a strain of COVID-19 virus. Therapeutic proteins like interferon. So interferon is a antiviral drug. We use recombinant E. coli to produce interferon. Yung insulin, this is a drug for diabetics. We use recombinant E. coli, to produce insulin. Monoclonal antibodies or MAB are produced using hybridoma cells. These are cancer cells, so they reproduce very fast. And then antibiotics, for penicillins, we use Penicillium chrysogenum. 330,000 to 400,000 tons per year. Cephalosporin are produced using Cephalosporium acrimonium at 100,000 tons per year, and tetracyclines is we use Streptomyces aureopacins to produce tetracyclines. These are different antibiotics. Okay, so sa board exam, you need to remember what bacteria or microorganism is used to produce the following product. So for example, nata de coco, ano ba yung ginagamit na organism? Vinegar. So, halimbawa, vinegar is acetobacter aceti, ethanol, Saccharomyces cerevisiae, soy sauce, so it's the soje, the bacteria, cellulase, trichoderma rizae. So you need to remember, the organism that produce these biochemical products. Yon. So nata de coco, if you still remember this from your industrial microbiology lab, we use Acetobacter xylinum, to produce nata de coco. Vinegar is Acetobacter aceti, ethanol is Saccharomyces cerevisiae, and soy sauce is Aspergillus soja, while cellulase is Trichoderma rizae. We need to remember, the bacteria that produces the product. Or on the other hand, when you are given the microorganism, you should be able to identify the product it produces. So for example, Saccharomyces cerevisiae produces ethanol. Penicillium chrysogenum produces penicillin. Trichoderma rizae produces cellulase. Aspergillus niger produces citric acid. No? Lactobacillus casei produces lactic acid. It sounds like lactic acid, di ba? Okay, so these are the four, these are the four branches of science, that is involved in biotechnology: microbiology, biochemistry, genetics, and biochemical engineering. So kayo, wala kayong micro, wala kayong biochem, wala kayong genetics, ito lang yung meron. Kami nung college kami, may micro kami, tsaka may biochem kami. Pero ang gagawin natin dito, i-discuss natin yung biochem before the midterm, Okay, so just try to remember these four science involved in biotechnology: microbiology, biochemistry, genetics, and biochemical engineering. What is the importance of biochemical engineering in biotechnology industry? Ano bang function niya? So biochemical engineering is one of the major areas of biotechnology important to its commercialization. So pag gusto na natin mag-produce ng product in the industrial scale, doon pumapasok yung biochemical engineering. So pag i-scale up, successful commercialization of biotechnology requires the development of a technologically viable and economically efficient process. So dapat, halimbawa, pag sinabing technologically viable, mataas yung kanyang yield. And then pag sinabing economically efficient naman, dapat hindi kayo malulugi. Dapat profitable. Okay? So ganoon naman talaga. Pag nagpa-plant design tayo, ito yung gusto natin mangyari. Especially the economically efficient. Yan yung sinacheck natin. Kaya tayo nag-feasibility study. So what does a biochemical engineer do? The biochemical engineer is a process engineer whose role is to transfer the knowledge of biological scientists into practical operation. It requires integrated knowledge governing biological principles and of chemical engineering methodology. So what is the role of biochemical engineer for commercial realization of biotechnology? So unang-una sa bioreactor. So usually sa laboratory, gumagamit sila usually mga 1 liter lang na bioreactor. Pero bioreactor pa rin tawag doon. So chemical engineer, yung knowledge natin, ginagamit natin for scaling up. So usually sa lab, batch lang yan sila. So pwede tayo mag-design ng mga PFR o kaya mga CSTR o kaya mga PBR reactor that will still produce the product. Tapos tayo rin yung mag-i-specify nung control at saka kung paano siya ino-operate. Kung ano yung flow rate, kung ano yung i-maintain na temperature, ano yung i-maintain na pH, paano ko kontrolin yung pH, paano ko kontrolin yung temperature. So, ano yun ng biochemical engineer? Duty yun. And also sa downstream processing equipment. Pag sinabing downstream processing, ito yung purification ng product. So i-specify tayo ng equipment to purify the product. Halimbawa, filtration, halimbawa, distillation, o chromatographic column. Pati yung sizing nun, tsaka yung design nun. Of course, the fermentation plant design. Ano? So mamaya pag-aaralan natin, usually dalawang type kasi yung fermentation tank, yung bioreactor. Merong isa seed fermentor, tapos yung isa yung main fermentor. So specify din natin yan. Ano? So next, we have here an overview of the biotechnological process. Ano? So una muna, we have the raw material. So this refers to the medium and the nutrients. Ano? So halimbawa yung gelatin, yung agar, di ba? Tapos yung mga nutrients na nilalagay doon sa dinidissolve sa agar. So dapat kasi may kumpleto yung kakainin ng microorganism doon para makapag-produce ng product, no? So ino-optimize yun para hindi mamatay yung bacteria, tapos piniprepare. Tapos yun namang microorganism na ginagamit, syempre ina-isolate yun at saka ini-screen. Tapos another thing na ginagawa doon is strain improvement. So nagpo-produce sila ng strain na mas mataas yung yield, ano? And then they isolate that strain. Tapos in this part, palimbawa na-produce na natin yung media, susunod doon is sterilization. Pag paghalo na natin, natunaw na natin yung media sa agar, ang susunod doon is sterilize natin, ano? para yung pag nag-inoculate tayo, pag naglagay tayo ng microorganism natin, siya lang yung magsusurvive doon sa media. So we need to sterilize the media. Yung mathematics ng sterilization, pag-aaralan natin later sa midterm, ano? Where we're going to use Jean Copley's. Pero sa ngayon, ang pag-aaralan lang natin yung rule of thumb, yung basic sterilization. Yung 121 degrees Celsius for 15 minutes, no? Tatandaan niyo lang muna yan. Tapos yun. So pag na-sterilize na, ilalagay na yung media sa bioreactor. So ang duty ng chemical engineer sa bioreactor, siyempre isa-size natin yan. Gaano ba siya kalaki? Tapos paano ba i-adjust yung pH niya, tsaka yung kanyang temperature, no? Paano siya i-operate, anong flow rate. Tapos ng bioreactor, we have the downstream processing equipment. So ano yung mga gagamitin nating equipment para ma-purify? Kailangan ba natin ng distillation column? Kailangan ba natin ng filtration system? Kailangan ba ng chromatographic column? No? Specify na natin. Tapos i-design na natin yung downstream processing. And then you're going to have the product. So ito yung schematic overview, no, ng biotechnological process. Yung mga activities na ginagawa. Okay, so this is basic steps in cultivating microorganism. First, you have to prepare the media, letter A, and then you need to sterilize. Tapos i-inoculate nyo na yung microorganism dun sa media. Tapos we'll start fermentation in the reaction vessel in the bioreactor. And then final step is the downstream processing or purification process. Okay. Doon tayo sa medium preparation. So generally may agar, tapos doon dinidissolve yung mga nutrients. Ano? You heat it, you boil it para ma-dissolve yung mga nutrients. And then there are two types of media, no? Yung mga ingredients sa ginagamit sa medium preparation. So natural media at saka synthetic media. Pag natural media, hindi alam yung chemical composition. Like beef extract, no? Yeast extract. Hindi alam yun, ano? Yun namang mga synthetic media, chemically defined composition, consisting of chemically pure and known organic or inorganic components. So for example, glucose, known amino acids like glutamate, synthetic media yun kasi alam natin yung chemical structure. Okay? So, you must be able to differentiate between natural media and synthetic media. Okay, let's take a look at the parts of a bioreactor. Now, let's start at the top. So, may steam tayo. Pumapasok yung steam, ano? So, maaaring dalawa yung purpose ng steam. Pwedeng sa sterilization, pwedeng sa adjustment ng temperature. Tapos may viewing port dito. So, kung nakapag-chem-eng lab na kayo, probably nakakita na kayo ng viewing port sa boiler natin, ano? So, parang glass ito, tapos makikita niyo yung loob. Yung sa boiler, kung nakapag-operate kayo ng boiler, makikita niyo yung apoy doon sa loob ng boiler. Pag sumilip kayo dito, ano? And then we have the impeller. This is used to mix the media, no? Then we have cooling jacket. So usually, as you can see, may pumapasok ditong cooling water para pag tumaas yung temperature, mapapalamig natin, ano? Kasi kailangan natin i-maintain yung optimum temperature. So pwedeng meron din ditong steam ulit, no? So pwedeng ito yung mag-maintain ng temperature, pwede rin siya sa sterilization. Tapos, halimbawa gusto niyo lang kunin yung laman ng media, dito siya wini-withdraw sa harvest. Ano? So you can also introduce air kung aerobic yung fermentation. And then sparger yung tawag sa nagpo-produce ng air bubbles. And then ito yung broth natin, ano? Ito yung media natin. This is where the cooling water goes out kasi pumapasok dito sa part na to. Yung gases, dito naman lumalabas. Halimbawa kasi ethanol, ano, may carbon dioxide, so dito lumalabas yung gases para hindi tumaas masyado yung pressure ng reactor. And then we have to maintain the pH. So meron tayong pH controller. So nag-a-add siya ng acid o kaya base. And then we have the motor. So this motor, syempre siya yung nagpapaandar sa mga impeller. Okay, so sunod, after medium preparation, this is the synthetic media versus the natural media. So as you can see, ito yung mga example ng synthetic media. Glucose. Okay, alam niyo bang i-draw ang structure ng glucose? Up, down. Tandaan niyo pa from your organic chem. Sucrose. Ano ang monosaccharides ng ano? Sucrose. Glucose at saka ano pa isa? Fructose, no? Tapos ammonia, NH3 lang yan, ano? Amino acid, depende sa amino acid, ano? Anong pinakasimpleng amino acid? Glycine. Anong R ng amino acid ng glycine? H lang, hydrogen lang, di ba? Of course, you have amino, NH3, and then the acid is COOH. Urea. Alam niyo naman yung structure ng urea, di ba? You have NH3 sa gitna. Tapos you have carbon, and another carbon on the other side of ammonia. Ano? Okay, basta alam niyo yung structure nito. Yun lang naman yun. Pag synthetic media. Pag natural media, hindi niyo alam yung exact chemical structure. For example, molasses. Molasses, ito yung juice ng sugarcane, di ba? Siyempre, pag in-extract niyo yung juice ng sugarcane, hindi lang naman sugar yung nandoon. May water pa siya, etc. etc. Ano? Corn steep liquor. Siguro ito yung na-extract sa corn, ano? O pagkatapos niyo siyang i-ferment, ito yung nakukuhang juice sa corn. Coconut water, sabaw ng coconut. Hindi natin alam yung chemical structure niya. Ano? Yeast extract, beef extract, so hindi natin alam yung chemical structure niya. Ano? And also peptone. Not really. Let's please check peptone online. Tanong kaya sa exam niya. Okay, so I hope you can distinguish between synthetic media and natural media. Next is sterilization. So sterilization is to destroy the contaminant organism. Gusto natin kasi yung organism lang natin, Saccharomyces cerevisiae, siya lang yung mag-grow dito sa media. Siya lang yung kakain ng mga nutrients dito. Otherwise, sayang kasi yung media. Kasi pag mayroong ibang organism dyan, masasayang lang yung mga nutrients. Kaya ini-sterilize muna natin before we add the microbiological agents. Ano? So usually ang ginagamit dito ay pressure cooker or autoclave. Ang rule of thumb, ito kailangan tandaan nyo, is 121 degrees Celsius, 15 minutes. Itong 15 PSI, ito yung pressure sa pressure cooker. No? 15 PSI gauge yan. So mga 30 PSIA, 15 PSIG. So, pag large scale, you inject steam through the jacketed bioreactor. Steam yung padadaanin natin dito ano during sterilization. Tapos dun lalabas. Okay, so let's start with let's discuss inoculation. Inoculation is the process of adding the microorganism to the media after sterilization. Seeding the culture vessel with microbial material. Must be done aseptically in pure culture. Okay? So large scale progressive inoculation with seed fermenters. So ito yung mga seed fermenters natin, no? Ito yung mga seed fermenters natin. So this is ang kaibahan ng seed fermenter sa main fermenter. Ito optimize siya for the production of the microorganism. No, pinapadami dito yung microorganism. Dito naman sa main fermenter, this is optimized to produce the product. Okay? So we started with a slant. Ito yung slant, ito yung test tube. Dito yung ating culture, no? Usually we get this from microbiological banks like halimbawa biotech, nabibili yan doon, ano? Tapos papadamihin natin yung zinc plus, tapos pag marami na sila sa plus, saka ita-transfer din sa seed fermentor. Tapos pag ita-transfer nyo na ulit sa mas malaking seed fermentor, ang rule of thumb is 5% to 10% of fermentation working volume. Halimbawa itong second fermentor natin, 100 cubic meter. So lalagyan natin siya ng 5 to 10 cubic meter na inoculate, no? Na galing dito. Okay? Okay, so after the inoculation, next is fermentation. So we're going to maintain the optimum temperature and pH in the bioreactor for some time. Pwedeng a week, pwedeng a day, depende sa product na piniproduce natin. So bioreactor is monitored to ensure the fermentation proceeds efficiently. So, mini-maintain yung pH, kung ano yung optimum pH, mini-maintain yung temperature, kaya may cooling water tayo, may steam, for a specific number of days. And then finally, the last step is the downstream processing. So basically, downstream processing refers to the purification of the product. So, halimbawa, we can use centrifuges, no? Discuss natin to sa di ba? Kung sa akin kayo nag-momentum transfer. So, pag imagine niyo pag sinentrifuge niyo yung broth, mabigat yung cells. So, may iwan yung cells dun sa centrifuge. Lalabas yung liquid. So, that's one way to purify the product. Halimbawa, ang product niyo single cell protein, di ba? May iwan siya dun sa centrifuge. Pwede rin yung mga product separation and purification equipment like filters, no? Chromatographic column, distillation columns. These are example of downstream equipments. Okay, so these are the useful concepts and terms for the board exam. You need to match the fermentation product with the microorganism. Halimbawa, ethanol, ang fermentation product. Anong microorganism na nagpo-produce ng ethanol? Saccharomyces cerevisiae. Cellulase, ang product. Anong microorganism na nagpo-produce ng cellulase? Trichoderma raceae. So, halimbawa, ang fermentation product ay penicillin. Ang nagpo-produce ng penicillin ay Penicillium chrysogenum. So, dapat memorize niyo yan. O kaya naman, vice versa. So, halimbawa, yung microorganism yung given, dapat alam niyo yung fermentation product. Halimbawa, Acetobacter aceti, ano yung fermentation product? So, vinegar. O kaya acetic acid. Ganun. Yun yung lumalabas sa board exam. Ano ibig sabihin ng downstream processing? Downstream processing refers to purification of product. So, ano ba yung examples nito? Halimbawa, distillation column, filtration column, centrifuge, ano pa? Chromatographic column. No? Basta purification. This refers to the purification of the product. Synthetic media versus natural media. So in synthetic media, alam natin yung chemical structure. Diba? So, halimbawa, glucose. kaya specific amino acid, o kaya ammonia, no? Synthetic media yon. Yung mga natural media naman, yung unknown yung chemical structure. Like yeast extract, yung uh, yung um, yeast extract, no? Yung beef extract. Okay? Hindi natin alam yung chemical structure. Okay, so dapat alam nyo itong dalawa, i-distinguish yung dalawa, yung synthetic media at natural media. Pure culture versus mixed culture. So, yung pure culture, pag isang bacteria lang yung present sa culture, halimbawa sa Saccharomyces cerevisiae lang, ano? So, pure culture yun. Tapos pag halimbawa, yung sa Saccharomyces cerevisiae lang, dinagdagan niya pa ng another bacteria para din ma-process yung mga hexoses, that's a mixed culture. Seed fermenter versus main fermenter. So yung seed fermenter, optimized to produce the microorganism. Yung namang main fermenter, optimized to produce the product. Anong rule of thumb sa sterilization condition? 121 degrees Celsius, 15 psi, 15 minutes. No? Anong rule of thumb sa inoculation size? 5 to 10% of the fermenter volume. Okay? So that's it. That's our introduction for biochemical engineering. So see you on the next video.